So welcome again friends. So welcome in the second video of our recombinant DNA technology video lecture. So in our first video I have talked with you a very big picture regarding recombinant DNA technology, where we have talked about why recombinant DNA technology is important by simply giving the overall process, right? That how from a human chromosome we can amplify a small target DNA sequence with the help of RTT in a host cell, if we are going for cell based DNA cloning, or if we are interested in cell free DNA cloning, so we can also go for it. So in that video I have told you that there are many enzymes involved in the overall process. So today we are going to talk about those enzymes. So I want to divide this whole enzyme series into several parts. In the introductory video, I will not only talk about the basics in what four categories the all enzymes involved in recombinant DNA technology are divided. I am also want to talk with you a brief description about template dependent DNA polymerase. That is the first enzyme involved in recombinant DNA technology. Okay, and so we will have one video for introduction and template dependent DNA polymerase, and we will divide the nucleus, that is one of the most important part, into three video, where in first video we will talk about these four, and the restriction endonuclease one, that is the most important one, and CSIR net used to ask very problematic questions regarding restriction endonuclease. So we are going to give this one two videos, and this whole one video. So we will spend four videos, and after that, in case of ligase, in case of end modification enzymes, we will go through the whole thing within two, or if possible in one video. Okay, and we will do the ligase part, that is the last part, in one video. So our enzyme will take 1 plus 1 plus 2 plus, you can say, 2 plus 1, so 1 2 4 6 7. So I will take total near about 7 video to complete the enzyme topic. Okay, so the enzyme topic will be crystal clear, right? So without any wasting time, let's begin. So the overall enzymes that take part in recombinant DNA technology can be divided into four basic parts, or you can say broad categories. Okay, so the first one is our template dependent DNA polymerase, second one is nucleus, third one is end modification enzymes, and the fourth one is ligases. Okay, so first one is template dependent DNA polymerase. By simply hearing the name, you can I hope you can easily figure out what they are telling us, what they want to tell us. They simply want to tell us that template dependent DNA polymerase are DNA polymerase that have the ability to synthesize polynucleotide complementary to the already existing DNA or RNA molecule, that we can say the template of them. That means, suppose it is a RNA molecule or RNA template. So DNA polymerase or template dependent DNA polymerase specifically have the ability to synthesize polynucleotide that will be complementary of that RNA that is existing. Okay, so we have divided this template dependent DNA polymerase into four different parts. Let me zoom. Okay, that will be better. So the template dependent DNA polymerase, the first part is DNA polymerase 1. In DNA polymerase 1, we have three properties. So DNA polymerase 1, we can also call DNA polymerase 1 as Kornbach enzyme by the name of its inventor, Kornbach enzyme. Okay, so DNA polymerase 1 is also called as Kornbach enzyme. The DNA polymerase 1 have 3' to 5' exonuclease activity, that is our proofreading activity, 5' to 3' exonuclease activity, and 5' to 3' polymerase activity. So what they have 3' to 5' exonuclease activity, 5' to 3' exonuclease activity, 5' to 3' polymerase polymerase activity, and that is the main activity that DNA polymerase 1 required in case of recombinant DNA technology. We are going to see it in our future video, so you don't need to bother about that. So let's move on. Reverse transcriptase. So in reverse transcriptase, we all know reverse transcriptase is simply an enzyme that have the ability to synthesize DNA from RNA. If we remember our central dogma, that is DNA to RNA, RNA to our protein. So we have our DNA. Let me zoom, then it will be more clear. So we have our RNA from DNA with the help of what? Transcription. And DNA will again synthesize itself with the help of replication, right? And RNA to protein is facilitated by translation. Okay, so in turn RNA to DNA synthesis will be facilitated by reverse transcriptase enzyme, and the process will refer to as reverse transcription. Okay, so this template dependent DNA polymerase facilitated reverse transcription. So as a RNA directs the DNA synthesis, so we can say this enzyme is RNA directed DNA polymerase, right? Simple. And the second property is it synthesizes DNA from RNA, that I have told you. And this is most important, that reverse transcriptase enzyme was synthesized by Howard Temin and David Baltimore independently in the year 1975 independently, sorry, and they received their Nobel Prize for the synthesis of reverse transcriptase in 1975 in physiology and medicine. Okay, so this point is important for those who are willing to appear in GATE Excel, as they used to ask about inventors, but CSIR net does not bother about that. They do not ask about some inventors about their discoveries, but GATE Excel used to ask. So those who are willing to appear in GATE Excel must need to take this in their mind. So reverse transcriptase are those that have the ability to synthesize DNA from RNA molecule. Okay, that is why we can call it as RNA directed DNA polymerase, as a RNA directs the synthesis of DNA. Okay, so our last one is stacked DNA polymerase, and this is, for me, this is the most important one for template dependent DNA polymerase. So what stacked DNA polymerase is? First of all, this stacked DNA polymerase was isolated from Thermus aquaticus, which is a thermostable bacterium. So stacked DNA polymerase was isolated from Thermus aquaticus thermostable bacterium, and that is the reason why they can survive in temperature 72 degrees centigrade. The second thing is they can also be stable at the temperature above 90 degrees centigrade. So as the stacked DNA polymerase are isolated from Thermus aquaticus, so they can work easily at the temperature 72 degrees centigrade and can stable at the above 90 degrees centigrade. And the most important thing is that stacked DNA polymerase is used in polymerase chain reaction. We are going to talk about this polymerase chain reaction in details in our future videos. The most important thing is of the stacked DNA polymerase is that they have two properties. One is 5' to 3' exonuclease activity and 5' to 3' polymerase activity, but they lack 3' to 5' exonuclease activity. That was exhibited by our first enzyme in the template dependent DNA polymerase group, DNA Pol 1. So let me sum up. So template dependent DNA polymerase are the enzymes that have the ability to synthesize new polynucleotide complementary to already existing DNA or RNA template. They have three different subgroups. One is our DNA polymerase 1 that have 5' to 3' exonuclease activity, 5' to 3' polymerase activity, 3' to 5' exonuclease activity. This 3' to 5' exonuclease activity is lacked by our stacked DNA polymerase. Okay, and the middle one is our RNA dependent DNA polymerase, or we can say our reverse transcriptase. This reverse transcriptase have the ability to synthesize the DNA from the DNA from RNA, and the inventor for this enzyme are our Howard Temin and David Baltimore. That is why we used to say Temin and Baltimore invented the enzyme reverse transcriptase, and they shared their Nobel Prize in 1975 for the invention of that. So let me talk about the remaining ones. So remaining categories are nuclease, end modification enzymes and ligases. We are going to talk about them in our upcoming videos, so you don't need to bother about this. So I hope this video will be helpful to you, and if it is, please like my video and share with your friends and subscribe my channel. Thank you. Bye bye.